I will next go through each of the 6 classes of enzymes, giving an example reaction for each. The enzyme lactate dehydrogenase is an oxidoreductase which reversibly converts pyruvate and lactate using NAD or NADH, depending on which direction it is going. This example provides a very important point. The activation energy decreases for the reaction, but it decreases for the reaction in the forward or for the reverse direction. This will be very important when we start talking about enzyme regulation. The enzyme glucokinase was shown previously, but what we notice looking at it for a second time is that a phosphate group is transferred from ATP to the glucose molecule. Our products are glucose 6-phosphate and ADP. Notice also the magnesium, which is written underneath the arrow. The magnesium ion is a required cofactor in this reaction. ATP represents a second substrate. Also note that I have in this case drawn an irreversible reaction, which does represent reality and is a topic for a future chapter. An example of a hydrolase includes glucose 6-phosphatase. On this slide, I've abbreviated that G6-PAse. This is also an irreversible reaction which uses water, hydro, to cleave the phosphate group off of glucose 6-phosphate, yielding inorganic phosphate, PI, and glucose as products. Class 4 enzymes, or lyases, are probably the most confusing class of enzymes there are. I have chosen fumarase to use as an example to contrast with the hydrolase that we just talked about. Notice that in this case, water is involved, but in this case, there is no hydrolysis taking place. Instead, the water is adding across the double bond to yield an alcohol product. Thus, when we are examining reactions, we cannot just see water and assume we have a hydrolase. Lyases always involve a double bond. An example of a class 5 isomerase is phosphoglucoisomerase. I should perhaps show this with the sugars in a different way in order to make it a little simpler to see how these molecules are isomers of each other. But I call your attention to the fact that they are both 6-carbon sugars, and one of them is present in a 6-membered ring, and one of them is present in a 5-membered ring. Finally, the class 6 or ligase enzymes are enzymes that will be fusing 2 molecules together, ligating them. As an example, I chose citrate synthase as it is one of the reactions that is very important to the central metabolic pathway. I have been using some terms and now seems like an appropriate point to strictly define them. The substrate is the molecule which is acted upon by the enzyme. In chemical terms, it is a reactant. We do not, however, use the term reactant, as we have seen that many enzyme reactions are reversible reactions. That having been said, we do refer to the product of an enzyme reaction as a product. If you are troubled by this discrepancy, welcome. Finally, a term which is important that I have implied but not stated is that of active site. Active site is often a groove or pocket of the enzyme where the substrate binds and is converted to product. Figure 8.12 of your book nicely shows this. The groove in the dark blue enzyme where the substrate molecule, which is mostly green with some red and blue, binds. And they're also showing it rotated 90 degrees. We need to spend some time talking about coenzymes, and I'm going to spend a good bit of time talking about NAD, as it is perhaps the most important coenzyme in all of biochemistry. But oxidoreductases, because they catalyze redox reactions, always need a coenzyme, or else the enzyme would end up being redoxed. Recall that by definition, a catalyst is unchanged in the reaction. The three possible coenzymes that are used by oxidoreductases are NAD, NADP, and FAD, which are converted into NADH plus proton, NADPH plus a proton, or FADH2. You should not refer to NADH2 as a product. I earlier mentioned that most coenzymes are vitamin derivatives, and NAD is no exception. NAD is synthesized from what we refer to as niacin. The structure shown here, which we call niacin, shows us that we should be calling it nicotinic acid. However, my understanding is that historically nicotinic acid sounded too much like nicotine, and authorities were concerned that people would stop taking their vitamins if it sounded too similar. Niacin is converted by an enzyme to nicotinate mononucleotide, NMN for short. Recall from basic biology that a nucleotide is composed of a base plus a sugar plus phosphate. Here we have niacin as the base, ribose, the 5-membered OH containing ring, as the sugar, and the phosphate. Another enzyme converts the nicotinate mononucleotide into desamino NAD. NAD stands for nicotinamide adenine dinucleotide. So here we have two nitrogenous bases, the niacin, the adenosine, two sugars, both ribose, and two phosphate groups. The prefix des refers to without. Finally, yet another enzyme uses glutamine and ATP as co-substrates to add the amino group onto the nicotinic acid moiety in order to form nicotinamide. NAD is used to degrade molecules in metabolism. It is not a prosthetic group but rather it diffuses in and out of the active site. There are two stereospecific forms of NAD which are highlighted in magenta on this slide. Note that we are going from an sp2 hybridized carbon in NAD to an sp3 hybridized chiral carbon in NADH. The hydrogen can be attached either to the front of the ring or the back of the ring, depending on the enzyme. And thus the hydrogen that is added will either be pointing forward out of the plane of the ring or behind the plane of the ring. Experimentally, an important feature that allows the study of oxidoreductases is that, as shown in this graph, NADH has an absorbency at 340 nanometers, whereas NAD does not.